Okay, so the last video we discussed packaging of DNA and a lot of things. Today our topic is evidence of a DNA as a hereditary material. Quite interesting. We'll see experiment done by the scientists, evidence of DNA as a hereditary material, because we know DNA is hereditary material which carry out of information from parents to offspring. So we'll see how that experiment was conducted, what was the main reason and everything behind it. So let's start. First thing, there was a first scientist which was the Griffiths experiment. Basically, I'm telling you the name, which was the Griffiths experiment. Otherwise, the scientist name was the Friedrich Griffiths experiment. Okay, let's check it out. First thing, he was a British scientist and he did this experiment on the mice, right? Okay, so let's start. He took two strains. Strains are simply kind of part which he took to make sure to check the experiment. So he took rough strain. Second was smooth strain. A smooth strain which is also like pathogenic, kind of harmful, and this one rough was non-pathogenic. I hope I am not writing completely well. What he observed that here is this kind of a structure. What will happen here? He took mice and inserted kind of micropolysaccharides, small small polysaccharides. Micro means small. A small polysaccharide which was taken. What he observed that these mice dies for no reason. I mean, they died for what? Because they were having what? They were facing pneumonia. Pneumonia happened because of the bacteria, pneumococcal or something bacteria. So he observed the insects or mice, they all die. Here what he observed even when they are adding any uh, micro polysaccharide, nothing was happening. They were quite happy. Mice were happy. Mice happy, right? So he observed what exactly is happening. He took the first step, which was about this one, identifying what really happened. Then comes the second one, what he observed. Secondly, he observed that was that heat, the surrounding heat killed strain. He took heat killed strain and then he injected in a mice. He injected that in a mice. Then thirdly, what he observed, like lastly, he observed that the heat killed a smooth strain plus at the time live RNA was added, like heat kill a smooth strain plus live R strain, like rough endoplasm strain was taken and was injected in a mouse. So what he observed? He observed that the mouse become dead. Why is it so? If I just directly tell you, here we know uh, when we were adding rough endoplasmic, not rough endoplasmic, but the rough strain at the time, our mice was quite happy. Nothing happened till here. A smooth endoplasm, not endoplasm, sorry, but a smooth strain was added at the time. They were, of course, we were supposed, because they were supposed to be die, because S strain, smooth strain, make them die. But scientists thought, why not to add something like R strain, because R strain which protect. So we observed that is even going to dead, like there was mouse dead even when we added right R strain, right? So this means something wrong was going on. What they observed, that mice then injected, then that mouse which was dead, they saw that in the mice the blood live S strain, smooth strain was found. So after that they saw this principle that's called as a transforming principle. transforming principle they observe. So this means as genetic material, then they observe, this means there's something in S DNA, not DNA but the S strand, because that's why the big blood become live even though we just added R. So this means there was something wrong, something going on, right? So what this all this mean, transforming principle started, transforming principle happened, but at that time S which was genetic material, so we don't know was that RNA. Are you sure? I'm not sure. Neither it was DNA, neither it was protein. So this means this all information was given by Griffiths experiment. Of course, this is kind of unknown description, so we need someone who could lead it out more. So then we had different scientists. Who were they? Okay, let's there were few more scientists. Who was Oswald? So we call this experiment as Oswald, Avery, uh, Colin McCree. There's so many scientists who done this one. McLeod, and the last one, Medor McCarthy something. I'll just read it out from the paragraph. These all scientists, what they observed, they literally come with the mind blowing experiment. What they saw that heat, which was going to kill, right? We know heat which would be killed by what? If we were adding live R. If we are adding live R, they were looking that there was nothing and no protein was formed. So if no protein added, this means there was not anything which could harm. This means there was no protease. Protease is simply enzyme which digesting enzyme, protein digesting enzyme. This means they observe that was not live R because no protein was formed. This means there would be no anything like that. This means then they come with another, which is was about heat killed, and they added live R. Live R, what they observed there was also no any concern, nothing happened. Why? Because here they added RNAs. We are observing was it protein? Now we are sure no, it was not protein. Then we added RNAs. RNA is a genetic material. A is when we are, this means we are discussing something related to enzyme. So RNA digest enzyme, which was this one, and when we saw there was no any concern. This means no, nothing was formed. Then in the last, here comes heat kills. What they observe here they added something more, which worked down. They added live R. Everything was same, but they were only checking which protein, which RNA, DNA. Here they added DNAs. DNAs, the one who eat DNAs, right? DNAs. This means they are enzyme which digest DNA. What they observed something interesting. This means there was not any conversion, no conversion to S live. Then what they observed, this transforming principle was DNA. Till last the Griffiths experiment, we were not known what transforming factor is there, but till now they thought, yeah, this transforming principle is nothing but the DNA, right? Because after that the protein was formed. So even after that one, not everyone was known what exactly is DNA. No one was known DNA. So this means no one find more about this experiment. Till this one it was right, but no one wanted to proceed more because DNA was kind of unknown thing at that time. Not everyone was known, right? So I hope it will be very much clear main concept of this experiment. Then let's check it out what's written in the book, because so many points different and interesting. Okay, evidence of a DNA. Then we, yes, like that factor, we got to know how DNA is formed. So Streptococcus pneumonia, which is a bacteria which causes pneumonia, that was found in two strands. Smooth, which is capsulated smooth form, S type. Second is non-capsulated, which is R type, right? So what they observe, Friedrich designed some experiment. During the course, he injected R type to the in laboratory to the mice and observed that no, like there was no any effect if R type was added. Then if they added S type, there was a fatal, like that means uh, proved fatal for mice. They observed that if both strains are heated, both killed at high temperature. Here's the experiment. They saw live S strain was added. Here live R strain was observed. So result was mice dies when we added S. Mice healthy when there was a live R, right? Then they did mouse was healthy, even bacteria cell found in them. Then we convert these and we saw mouse dies when the S strain found in the heart of the mice, right? So this means I have told you everything. So here comes the main part. So this means they were telling he designed some more experiment found about these one to find out the result. This experiment were concluded and have satisfactory explanation of the result. Then everything clear. This phenomena in which heat killed S type could have had the effect of R type. That's basically called transformation. Then nearby, like after 13 years, in 1944, basically, there were scientists Avery, McCarthy. They discussed the identifying material of the Griffiths. They tested this heat kill bacteria for the transforming, find out that it was not RNA or various protein but only the DNA which possessed transforming property. If the enzyme deoxyribonucleic hydrolase was added to the heat killed S type bacteria, so the transforming property was lost. S was added, transforming property was lost. Therefore, it has become clear beyond any doubt that DNA must be having some kind of genetic information. That's why it was able to do that. So today we can say evidence of DNA. How evidence is simply how exactly we found anything we are discussing. So here topic was about evidence of DNA. We know DNA is a hereditary material, but how we get to know that? We get to know that by the Friedrich Griffiths experiment first. But here until it was quite unknown till the end because he could not tell what exactly was that. He could not name it out either it was DNA, RNA, or protein. So yes, then comes the other scientists and they discussed all. So I hope it will be very much clear. Thank you for watching. Bye bye.